So I'm going to be talking about our recent work, really tying together neuroimmune control of allergic immunity. So the way that I want to start is I just kind of want to introduce, of course, why we care about this in the first place. And I don't think it's a surprise to anybody. We all know this. Atopic diseases are on the rise. And these statistics are from the US, but we see this across the globe. And this is really becoming more of a global problem than it ever has been before. We see increases in asthma, allergic rhinitis, and especially atopic dermatitis, a chronic itch condition that really can cause so much chronic pain or chronic issues for people. And then, of course, we see a huge increase in food allergies. And this is something, these rises in atopic dermatitis and especially food allergies are things that just aren't slowing down. And despite the fact that we have such a high proportion of our populations that are affected by atopic diseases, by allergic diseases, and despite the fact that these are increasing on a yearly basis, we have really no idea how they are started. Okay, we have no idea how these are initiated. And so that really has always been the focus of my lab, is looking at how allergic immune diseases get started in the first place so that we can better understand and hopefully target and prevent their initiation moving forward. So if we want to understand how they get started up, I want to look at where they get started, okay? And what I'm showing you here is just a little paradox, I think, for a lot of people, that when we think about allergic immune diseases, it's easy to imagine that atopic dermatitis, an itchy skin disorder, can start up in the skin. But what's really interesting is that we think that most allergic immune diseases, for instance, food allergies, actually get started up in the skin as well. And that information comes from a lot of epidemiological data showing that babies, when they get their first exposure to peanut, if they are to be peanut allergic, 70% of them have that anaphylactic reaction or that whole body allergic reaction on the first exposure to peanut. And where are they exposed to peanut? Well, it's interesting because if we look at exposures that happen in the skin, we know that being exposed to peanut protein and household dust increases your risk of developing a peanut allergy by 200%, okay? So doubles your risk, okay? However, if you're exposed early to peanut allergens, through the gut, through the oral mucosa, we know that you actually have a reduced risk. And so this has really, these observations have really set up our interest in looking at the skin as the site of preliminary allergic sensitization. So how does allergic sensitization happen? How does the immune response get started in the first place, okay? Well, it all comes back to simple innate control of adaptive immunity, okay? And anybody who's ever seen me give a talk has probably seen this slide because I've been showing it for years now. And what we know is that the innate immune system is able to take in information from pathogens. It's able to take in an input and lead to an adaptive immune system activation or the output. And the immune system, the innate immune system specializes that adaptive immune output based on what its input is. And that means that if it sees LPS, if it gets activation through its pattern recognition receptors, it leads to an appropriate Th1 or potentially even Th17 type output, okay? However, if the innate immune system sees a protease allergen or any other type of allergen, it recognizes that, likely through functional recognition, okay, leading to Th2 differentiation or an allergic immune output. But we know that this isn't really just happening in boxes. If we look in the skin, if we want to know how this is happening, we know that there's one cell that's really in the middle of all of this, the dendritic cell. And although probably about 15 years ago or so, we may have thought that one dendritic cell was everything for everybody, that one dendritic cell could tell the difference between LPS or allergen and could differentiate between those inputs to give you different outputs. We now know that, that dendritic cells are much more specialized than that. And if we look at the skin of a mouse and zoom in, What we know is that we can differentiate dendritic cells based on all of our terminology, CDC1, CDC2s, but we can also differentiate them based on transcription factors, and we can differentiate them based on surface markers, okay? And the surface markers that we used in the paper and in the data that I'm going to be talking about are CD103 positive DCs and CD301b positive DCs, okay? These CD103 positive DCs are a mixed bag. They include DCs that can induce Th1 and Th17, whereas the CD301b positive DCs, which were originally described by Akiko Iwasaki's group, are specialized in being able to induce Th2 differentiation. You get rid of these, you lose Th2 differentiation. So this sets up a really nice way for us to specifically look at the signals and the triggers that will induce activation on this side of our CD301b positive DCs and migration, which is tied ultimately to Th2 differentiation, okay? So if we just want to take a step back and see what this looks like in case you don't usually play with DCs, what you'll see in the response to a type 1 stimulus like LPS is you'll see that the CD103 positive DCs here will upregulate activation markers. Here I'm showing you one that's called PDL2. So they'll upregulate the expression of activation markers, which gives you evidence of DC activation. And then that is coupled with DC migration through the upregulation of CCR7 expression. And what you can see is that LPS will induce specifically the migration of CD103 positive DCs into the draining lymph node. And if we look at the response to protease allergens, we see basically the same thing, but this time for CD301b positive DCs. So now we see that protease allergen exposure, in this case we're using Papain, induces the upregulation of this activation marker PDL2, you can see it right here, and it leads to the specific migration of these CD301b positive DCs into the draining lymph node. So it looks like DCs sense the stimuli, and then that leads to activation and migration. But there is a dirty little secret in DC biology, especially when it comes to type 2 immunogens and allergens. And this is what got us excited and led to the paper that we're going to be discussing. So if we look at DCs and their response to in vivo immunization to LPS, again, that Th1 type trigger, or Papain, again, that Th2 type trigger, we see that DCs, again, are able to upregulate PDL2. I just showed you this. This is old news, okay? And again, they're able to start migrating. This is chemotaxis in a dish, but they're able to start moving after they were exposed to Papain in vivo or LPS in vivo, okay? So they're activated, and it looks like they're functional. They can migrate, right? But what's really interesting, and what is that dirty little secret that I told you about, is that what happens in vivo is very different from what happens in vitro. So in vitro, if we take DCs and we stimulate them with LPS, we see the exact same thing that we see in vivo. We see upregulation of maturation markers, they look activated, and we see that they begin to migrate, okay? They seem to be functional. But what's so interesting, and what has always, I think, dogged the field, is that when it comes to allergens, okay, and in this case we use Papain again, they have no effect on DCs in vitro. So here we have DCs that were stimulated with Papain in vitro. They do not upregulate activation markers. They do not migrate. It's as if they couldn't sense it at all. And this led us to the original question that we wanted to really look at, which is, how are these DCs sensing allergens in the first place, okay? Actually, are they sensing allergens in the first place? Or potentially, could there be a sensor intermediate to the allergen and the DC that interprets that signal and passes it on to the DC? So if we were to think of a sensor, and we're a cellular immunology lab, I like to think in terms of cells, we thought of certain qualities that that sensor would have to have. And the way that we kind of divided it up, we thought, well, quality one is that it would have to be present throughout the body with specific concentration in epithelia. It has to be at the right place to find the allergens, right? It has to be capable of detecting a wide variety of enzymatic activity. I'm not going into all the background, but my goodness, allergens are different and type 2 immunogens are different. They have different activities, they have different functions. They pretty much all share in common some sort of enzymatic activity, but it's so wide. So whatever is sensing them has to be able to detect, again, a wide variety of activities. It's got to be able to act immediately. We can't be waiting hours for the first sensor to activate and then hours for the dendritic cell to activate and migrate. And it really has to be independent of the adaptive immune system. Why? Because we're seeing this happen in mice that have never seen any of these allergens before. So we know that this is independent of the adaptive immune system. So what could actually tick off all those boxes? Well, a hint for us came from a very normal encounter. So I was out with my 3-year-old and this happened, okay? And if this has happened to you, you might know right off the bat that this is not any normal, ordinary hive. This is the result of a bee sting, okay, or a wasp sting, okay? And what we noticed, or what I noticed when my 3-year-old got stung, was that he was instantaneously in pain and he was itching. He was miserable, okay? And it was that instantaneous feeling of pain and itch that made me realize all of a sudden that potentially the sensor for allergens, because remember, venom from bees and wasps, whew, it is a powerful allergen. It's one of the most powerful allergens around, okay? So that made us realize that potentially the sensor was something under our noses the entire time. Potentially it was the peripheral nervous system. And I'd like to say that I was the first one to think of this, but neurobiologists have known this for ages, that venoms can directly activate sensory neurons, okay? Now, of course, this is important to get that stinging insect away, but we wondered if it was also important because maybe sensory neurons could be upstream of dendritic cell activation. And that's what we wanted to test, okay? So we wanted to find out if these allergens could be activating sensory neurons and passing that signal on to dendritic cells to initiate the whole allergic immune response. So to do that, or to ask that question, we first had to see if our allergen, papain, even induced a sensory response to begin with. So it's funny, the gold standard for doing this is actually a behavioral test where you inject a small amount of substance under the cheek of a mouse and look to see what the mouse does. If it picks up its forepaw and wipes with one paw, then it's showing you that it's in pain, okay? And this is a painful stimulus. However, if it picks up its hind leg and scratches, and I'm sure if you have a dog or a cat, you've seen your pet do this, that means it's itchy. And I'm gonna show you some examples of that right here. So hopefully you guys can see these videos. Right now, these are mice that were injected in the cheek. And what you can see is this mouse over here is picking up its forepaw and wiping. And this mouse back here is picking up its hind paw and scratching, indicative of wipes or pain, scratches, itch. And what we can do is we can quantify the number of wipes and the number of scratches and have an idea as to what sort of sensory response this animal is experiencing. And so when we did that, and we compared allergens like papain and alternaria extract to things like histamine, we saw quite surprisingly that on the first exposure to something like papain or alternaria, we were seeing an itch response that was equivalent to what we see with a pure itch-inducing stimulus, histamine. Interestingly, papain was a mixed response, just like we see with venom sometimes. Papain induced not only the itch response, but it also induced a pain response that was equivalent to what we were seeing with lower doses of capsaicin. So this indicated to us that we were potentially seeing direct activation of sensory neurons. This was happening in the first 20 minutes right after injection in naive mice. So we thought that we were seeing direct activation of sensory nerves. But in order to verify whether or not we were actually seeing that, we wanted to go in vitro to do what we couldn't do in dendritic cells and do that with sensory neurons, see direct activation. So to do that, you can simply dissect out dorsal root ganglia and culture them. And when you expose those cultured dorsal root ganglia to papain, we saw we saw calcium flux, okay? So we saw evidence that the neurons were being activated by seeing this calcium flux. And we saw that papain induced a nice, brisk, and acute calcium flux. And what was interesting is that although it didn't activate all neurons, we shouldn't expect it to, it activated about the same percentage of neurons that were activated by histamine, really indicating to us that this might be a specific response, or it might be activating a specific subset of neurons. To understand if it was specific, we looked to see what the papain responsive neurons also responded to. And so we looked at whether or not papain responsive neurons responded to histamine, chloroquine, which is another itch-inducing stimulus, or capsaicin, okay? And what we saw, interestingly, was that capsaicin responsiveness was most enriched in the papain responsive neurons, okay? Now, it wasn't perfect, okay? Not all papain responsive neurons also responded to capsaicin, but it indicated to us that we were mostly seeing a TRPV1 positive neuron responding to this allergen, okay? So to prove whether or not TRPV1 neurons were responsible for this, we took two approaches, okay? First, we used a pharmacological inactivator, QX314, which is a lidocaine derivative that's supposed to be pretty specific for TRPV1 neurons, okay? And we co-injected it with papain. And what you see is that co-injection of this QX314 agent really blocked the itch response to papain, also blocked the pain response as well. And then to get at it a little bit more specifically, we depleted mice of TRPV1 positive neurons by using TRPV1 DTR mice that were treated with diphtheria toxin. And when we looked at their itch response to papain, we saw again that we lost our itch response. So now when we go back to the original model, we're seeing that protease allergens certainly seem to activate TRPV1 positive sensory neurons, okay? The question is, does this have anything to do with DC migration? Because that's ultimately what the hypothesis was. Well, first, to see if it was even possible, we did immunofluorescence to see if the dendritic cells were even at the right place to respond to the sensory neurons. And so what you can see here is skin from the naive ears of mice. And you can see neurons that are dual labeling with TUJ1, which is a pan-neuronal marker, as well as NAV1.8 cherry, which is marking this sensory neurons or nociceptive neurons in the mouse. And so what I hope you can appreciate is that our DCs marked by CD301b and MHC class II dual expression are really tracking along the neurons in the skin. And interestingly, it's not just that all DCs track with these neurons. It really seems to be specific to this subset, because if we compare their localization to neurons with the other DCs in the skin, we see that other DCs are located further away from neurons, but these DCs, these type 2 or Th2 skewing DCs, are really in close contact with neurons. So these DCs are in the right place at the right time to respond to neurons. So do they? Okay. To answer that question, we went back to our QX314 pharmacological inactivation of these neurons and looked to see whether or not we could silence these sensory neurons with QX314, whether or not that silencing would lead to any effects on the DC. And what's nice about this approach is that it would leave all other possible ways of activating DCs intact. It would leave all the tissue damage. It would leave all the potential alarmin release intact. But this way we would take the neurons out of the equation. And so we use the KD mouse model where you can photoconvert the skin and really track the movement of skin resident DCs from the skin to the draining lymph node. And by doing that, we compared DC migration in mice that received papain versus mice that had received papain in the presence of QX314. And what I think you can see in both graphs that are showing percentage of migrated CD301bs as well as total number, that these CD301b DCs did not migrate if the TRPV1 positive neurons were inactivated by QX314, okay? What's interesting is that this was really specific to the CD301b positive DCs and allergen exposure. If we look at the migration of other DC subsets in response to things like LPS, it's completely intact, completely unaffected by the inhibition of the TRPV1 positive neurons. So then we wanted to look at it a different way, again, using our TRPV1 DTR mice now to actually deplete these neurons, get away from the pharmacological inactivation. And so using the same KD type model, what we were able to show was that depletion of TRPV1 positive neurons led to a loss in the migration of CD301b positive DCs, again, by both percentage and number. So together, these data really put the TRPV1 positive sensory neurons upstream of CD301b DC activation. But how were they interacting? How are they talking to each other? Well, we certainly know that sensory neurons in the skin can release a lot of neuropeptides locally to activate cells. And we know that the release of CGRP has been shown to activate CD301b positive DCs to induce an antifungal pathway. That's been shown by Dan Kaplan's group. We also know that substance P release can directly activate mast cells, okay? And that can lead to itch responses. And so we were really interested in both the CGRP and substance P pathways. And so we looked to see whether either of these neuropeptides were released in response to allergen. And whether or not we looked at flank skin explants or whether or not we looked at dorsal root ganglia cultures, we saw the same thing. Allergen induced a robust release of substance P, shown here, okay? And it inhibited the release of CGRP. That pretty much set up nicely the next hypothesis, which was that substance P was inducing DC migration. So to examine that, we went back to our KD model, and this time injected mice, yes, with just a simple OVA sham immunization, Papain to induce migration, but also this time with CGRP and substance P. And what I think is very clear here is that substance P was able to induce robust DC migration, specifically of the CD301b positive DC subset. So you see them migrating amazingly in response to substance P with no effect of CGRP, okay? And CGRP, interestingly, did not have any effect on 103s either. So now we're growing our model. That TRPV1 positive neurons get activated, they release substance P to activate the CD301b positive DCs, but how is it activating them, okay? To try and understand that, we looked at the receptors that were expressed on these DCs. Now, TACR1 and TACR2 are the stereotypical substance P receptors, but recently MRGPRA1 and MRGPRB2 have both been described to bind to substance P. So we looked at expression of all four receptors and found on bone marrow-derived DCs that MRGPRA1 was specifically expressed, and saw again in our primary DCs from the skin that CD301b positive DCs showed MRGPRA1 expression, whereas the 103 positive DCs did not. So to really ask whether or not MRGPRA1 was the functional receptor for inducing this dendritic cell migration, we made bone marrow chimeras where we were able to completely make all of the CD301b positive DCs MRGPRA1 deficient versus wild type, okay? And comparing the migration of these DCs in both of those models, I think you can see very well that MRGPRA1 deficiency leads to a defect in DC migration. To look at it a different way, we did competitive bone marrow chimeras where we were able to transfer in a 1-to-1 ratio wild type versus MRGPRA1 deficient bone marrow into mice, then do a foot pad immunization to look to see whether or not the DCs from wild type versus MRGPRA1 knockout mice were able, or bone marrow were able to migrate better. And I think it's quite clear that the wild type were able to migrate much better to the draining lymph node than the MRGPRA1 knockouts. And again, what's interesting is that in response to LPS, there was no defect in CD301b DC migration. There was no effect of MRGPRA1 deficiency on these other DCs. So how is it inducing this? Well, if you want to think about how cells move, cells have choices. They can either just sit in place, they can be sessile, they can randomly move about. And in this case, if you put them in a transwell, some would fall through, but but many would also stay in place just because of that random movement. Or you could have chemotaxis, directed movement, where you would really see this increase in response to a chemokine ligand or a chemoattractant. To look to see what was happening after substance P, we looked at chemokinesis, where there was no chemoattractant, and we looked at chemotaxis, where cells were migrating to CCL21. And what was interesting is that we saw that substance P induced this big boost in random migration of the DCs. Same with FMRF, which is a specific agonist of MRGPRA1. Now, this migration was boosted in response to CCL21, but you can see it was even greater than what we see with LPS. And since LPS induces CCR7 upregulation, and we know how important that is for migration, it just gives you an idea of what a powerful stimulus substance P really is. So we're putting together the whole story, but ultimately what we really want to know is if we mess up this pathway, will we lose Th2 differentiation? Okay. So to get to that, we again depleted our TRPV1 positive neurons using our TRPV1 DTR mice and looked to see what would happen with Th2 differentiation. And as you would imagine, based on our previous data showing that the DC migration is lost, we also lose Th2 differentiation. So TRPV1 DTR mice had less IL-4 production, IL-13 production. They had slightly less interferon gamma production, which we think is simply a result of less DCs making it into that draining lymph node. So in summary, we really have set up this new pathway for thinking about how DCs get activated by allergens. The first step is exposure to a protease allergen. And what I didn't show you is that we showed the same data with cysteine protease allergens, serine protease allergens. So this isn't just a one allergen type thing. But protease allergens activate TRPV1 positive sensory neurons, leading to their release of substance P, which activates CD301b positive DCs specifically through their specific expression of MRGPRA1, leading to chemokinesis, allowing them to start migrating around in the skin. And then they use their level of CCR7 expression to migrate towards CCL21 gradients, pulling them into the lymph node, where again, they use a little bit of CCL8 produced in the lymph node and their own CCR8 to pull them across into the lymph node to induce Th2 differentiation. So, you know, there are a lot of questions that come out of this, okay? You know, do all allergens activate TRPV1 neurons? How specific is this? Are TRPV1 positive neurons a central sensor of allergens? Can the immune system alter this in any way? Can we boost sensing by sensory neurons? Can we inhibit this sensing by sensory neurons? That's what we want to know if we want to treat allergic disease, right? Do neurons actively regulate DC numbers or function in homeostasis? What's happening? What's happening at steady state? And of course, what are the other factors that are required for Th2 differentiation? Because one other thing that we showed was that we can force DC migration, but it's not enough just to force DC migration with substance P. That's not enough for Th2 differentiation. There's still more that's necessary. And I'm going to be honest, we don't know what it is just yet. So these are questions that we're interested in that I think really kind of came out of this work. And then I just want to thank my fantastic lab over at MGH, our funders and our collaborators. Isaac Chu was a huge help. Xinjiang Dong provided all the MRGPRA1 knockout mice. Mark Hoon provided the TRPV1 DTR mice. And we got to have a lot of fun doing experiments that didn't make it into the paper, looking at DC migration with Daniel Uremia's group. So thank you guys so much for your attention.
Thank you very much, Dr. Caroline, for your great talk. So if anyone has questions in the chat, please type it and I will be reading. I think we have a first one from J Ortiz, and I will read it. It says, since TRPV1 is also expressed on immune cells, including T cells, have you recapitulated these results in a model where you are specifically deleting TRP1 from sensory neurons?
Yeah, Jay, this is a great question. And it is controversial. I'm going to be completely honest with you, because some people can detect TRPV1 on these different cell types and some people cannot. And the actual expression of TRPV1 is not super high. What I can tell you is that TRPV1 DTR depletion does not lead to depletion of any T cell subsets that we can detect at all. Another way to go about this, though, is to look at NAV 1.8, NAV 1.8 targeted mice. Now, these are tricky because they also lack a lot of pain sensation, so it's not as even specific as the TRPV1, but we can phenocopy what we see in TRPV1 depleted mice with NAV 1.8 CRE-targeted lines.
Okay, so we also have a question from Jerry Lin. He says, great talk. Do tissue-resident mast cells who can respond to substance P and IL-33 play a role in CD301b dendritic cell migration?
Yeah, that's a great question. So there was a fantastic paper, gosh, a year and a half ago, showing that substance P released by allergen activated neurons can lead to mast cell activation, leading to itch and local inflammation. What we showed is that, surprisingly to us, we don't see any effect of mast cells at all in our model. So we can use mast cell deficient lines and we have no effect on DC migration, and we have no effect on the initial itch response. So I think what substance P in mast cells is doing is really inducing this inflammatory response and this local inflammatory response on the first exposure, and that potentially with IgE cross-linking, then it's really playing a role probably with DC migration.
Okay, so I will go with the last question from Veronica. She says, have you been able to check levels of basal substance P or MRGPA1 expression on patients with full allergies? Because this could be used as a predictor of levels of allergic reaction.
Veronica, that is a fantastic question. And substance P levels can actually be looked at even systemically. And that is something that actually we are trying to get involved in right now. So